Any questions about aldosterone? And students often confuse aldosterone and ADH, and then ANP, which we're going to talk about now. So make sure you're keeping these things straight. Aldosterone is the one that's involved in sodium reabsorption. It's the one that's involved in the Rast system. ADH is the one that's involved with the posterior pituitary. It's the one that's involved in water reabsorption, with the aquaporin tubes, whereas aldosterone is going to be those sodium leak channels. So again, find a meaningful way for you to kind of separate these and not confuse them, which is often done. Okay, let's move on here to speak about atrial natriuretic peptide, or ANP. And this is a hormone that is secreted by the atrial cells in the heart, right? So the walls of the heart are another important place where we can measure or detect changes in blood volume, because the heart has to stretch and accommodate all of our blood volume. That's another really good place for us to be detecting any important changes in our blood volume. So in response to a distension of the atrial wall, we secrete this atrial natriuretic peptide. And what it will do is increase GFR. It's going to help dilate the afferent arteriole and constrict the efferent arteriole and increase our GFR in response to distension in the atrial natriuretic wall. So what it will do is decrease sodium reabsorption by closing those sodium channels on the apical membrane. So we talked about those leak channels that were on the apical surface of our principal cells in the distal tubule and collecting duct. And these cells are helping to reabsorb sodium. Now, if we have a lot of sodium existing already, right, because we have a lot of that stretch coming on the atrial wall, which is indicating higher blood pressures and higher blood volumes, what we're going to want to do is decrease the amount of sodium that is being reabsorbed. So we're going to close those sodium channels and allow more of that sodium to stay in the filtrate and be excreted as a part of the urine. Okay, so in this way, aldosterone and ANP are like evil twin brothers. They're antagonistic. Aldosterone wants to pull more sodium back into the vasculature. It wants to increase blood pressure. It wants to increase blood volume. ANP wants to send more sodium into that filtrate, into that urine, to be lost from the body. It wants to decrease blood volume. It wants to decrease blood pressure. And again, it is being sensed or detected by that stretch that's being exerted on the atrial wall. So the overall effect of ANP is to lose more sodium in the waste, to excrete more sodium as a part of the urine. Okay. So let's look at an example here, kind of a flow chart describing that. So the main stimulus for ANP secretion is higher plasma volume. So if there's increased plasma volume, which is also expressed as increased blood pressure, we're going to see ANP being released. So that will be detected by the walls of the atria. Remember, the atria are our volume chambers. They are our filling chambers. And so they're the perfect place to monitor or to detect if there's too much volume, if there's too much blood in the vasculature. So if we have a lot of stretch happening on the atrial wall, that is a stimulus for ANP secretion from those atrial wall cells. So they will secrete ANP, and ANP will help to decrease sodium in the plasma by increasing sodium excretion. And what it does is it's going to cause the afferent arterioles to dilate. It's going to cause the efferent arterioles to constrict. That will allow for increase in GFR. So if we open up the afferent arterioles, more blood can get through. But if we close down the efferent arterioles, we create more resistance. And again, more of that blood is going to go through the glomerulus. That will increase GFR. So that is going to help increase sodium excretion, just shoving more blood through the kidneys to be filtered. That alone will help lose more of that sodium and lose more of that fluid. The second thing that will happen is we're going to decrease sodium reabsorption. So we're decreasing sodium reabsorption by blocking those sodium cells in the kidney. So if we block those sodium cells, we are going to prevent that sodium from being reabsorbed, right? Remember those leak channels across the apical surface. If we close them, we can't move more of that sodium into the vasculature, and so it gets lost as a part of that waste in the urine. So that's kind of the second thing we do. We directly decrease sodium reabsorption, but we also decrease renin secretion, right? So we decrease renin secretion, which is going to decrease aldosterone secretion according to that Rast system. If there's less renin, less angiotensin 1, less angiotensin 2, less aldosterone. And having less aldosterone is an indirect way of decreasing sodium reabsorption, because we know that aldosterone wants to be absorbed more sodium. And if we have less of it around, we're going to lose more of that sodium in the urine. So we're both indirectly and directly decreasing the amount of sodium being reabsorbed by closing the sodium channels, but also by decreasing aldosterone secretion, which is going to help with that reabsorption. So all in all, the end goal of ANP is to increase sodium excretion, which is to decrease the amount of sodium in our plasma, as well as decrease the amount of fluid that's in our plasma, which will help to decrease our plasma volume. Okay, let's take another pause here and see what questions we have. So just to kind of recap what we just spoke about sodium's reabsorption, the mechanism across the proximal tubule, distal tubule. We then talked about how aldosterone influences that, how aldosterone influences specifically the way that sodium is reabsorbed across the distal tubule and collecting duct by manipulating those sodium leak channels. And if sodium can't get into the cell, it can't be reabsorbed. We then spoke about the antagonistic hormone of aldosterone. So that was ANP. And ANP is going to be seeking to work in the opposite direction. It's going to be seeking to lose sodium into the urine. So it's going to block those leak channels for sodium, but then it will also influence the amount of aldosterone. And because aldosterone wants to reabsorb sodium, if we decrease the amount of aldosterone, we also decrease the amount of sodium reabsorption. Okay. Another important thing we talked about was the Rast system. So really make sure you understand the Rast system. Renin coming from those JG cells, or also called granular cells. Renin stimulating the angiotensinogen to be converted to angiotensin 1. Angiotensin 1 converts to angiotensin 2 by ACE, so angiotensin converting enzyme, and then angiotensin 2 having several different responses, all in an attempt to increase mean arterial pressure. And the main response of those 4 responses is increasing aldosterone, which increases more sodium reabsorption and eventually results in increasing more fluid, which increases pressure. Okay, so those are kind of the important things that we just talked about. So let's look at these checkpoint questions here. All right, so let's look at question 1. So question 1 is to briefly explain how an increase in the secretion of renin stimulates the secretion of aldosterone. So I kind of went over that, so I may have jumped the gun there, but just explaining the renin-angiotensin system is what this question is asking. So who can summarize the flow of how do we get from renin to aldosterone? Any ideas? Okay, so in the interest of time, we basically have 4 steps. So, okay, excellent. So someone responded here. Renin binds to angiotensinogen, which starts the cascade. That's perfectly correct, right? That is absolutely correct. And then we go from angiotensinogen to angiotensin 1, angiotensin 2, and then we go to aldosterone. So secretion of renin is directly proportional to secretion of aldosterone. Okay. And the second part of the question, how does an increase in aldosterone secretion affect sodium reabsorption? So in short, will that increase or decrease sodium reabsorption? Increase, excellent. So aldosterone will increase sodium reabsorption. And just to reiterate the mechanism there, by inserting more of those sodium leak channels into the apical surface of the principal cells of the distal tubule and collecting duct. Okay, so really make sure that you drive in that specific location, the specific type of cells, and the surface of the cell, right? The apical surface, there are leak channels, so they're going to be moving sodium passively, and then that's how they get into the cell. They also help to insert more sodium-potassium pumps in the basolateral surface as well. Okay, so that will facilitate more sodium being moved across the cells, across those principal cells, and into those peritubular capillaries. All right, and then number two, where are the cells that secrete renin located? So they're in the kidney, excellent, yes, they're in the kidney. Not the adrenal cortex, but they are within the nephron, so kind of a hint there. So they're part of that complex that we talked about, but specifically, if anyone can remember where the granular cells or the juxtaglomerular cells were located. So if you remember the juxtaglomerular complex, something we talked about before, make sure you understand that complex. We had the macula densa cells. They are in the distal tubule, right? And we talked about how that distal tubule comes into very close contact with the renal pole. So it sits right in between the afferent and efferent arteriole. So these three cluster of cells are communicating with each other through these chemical signals that we call paracrine factors. So the macula densa were in the distal tubule. The juxtaglomerular or the granular cells were actually in the afferent arteriole itself. And then the mesangial cells, the third set of cells in this complex, were kind of stuffed in between the renal pole there. So specifically the afferent arteriole cells, so those juxtaglomerular cells, the granular cells that are in the afferent arteriole, these are the ones that are secreting that renin. Okay, all right. And then the second part of the question here, name 3 stimuli for renin secretion. So if anyone can kind of quickly recall those 3 stimuli. So what are the things that are happening that allow or send the signal for renin to be secreted? And we talked about this idea that renin kind of sets the whole cascade off. So the stimuli for renin are the initial stimuli that start that Rast system. So renin starts being secreted first, and then we have the rest of that cascade going. So what are the things that will signal for renin secretion? Decreased pressure in the afferent arteriole, excellent, yes. Decreased pressure in the afferent arteriole, that's one. And that would make sense, right? The afferent arteriole cells are the ones that are secreting that renin. So if there's decreased pressure coming through there, they're immediately going to start sending that renin, and then we're going to have that cascade going into effect. What were the other 2 stimuli? Renal sympathetic nerve activity, excellent. So that was the second stimulus, and that was through the baroreceptor response. So if we have less stretch coming on our baroreceptors, right, where they are located in the vessels, if you have less stretch there, that is going to stimulate the sympathetic nerve activity, which is going to clamp down on our afferent arteriole, increase the afferent arteriole, clamp down on the efferent arteriole, and help to correct that by increasing GFR. And then what was the third stimulus that we saw? Decrease in sodium and chloride in the distal tubular filtrate. Excellent. So if we have less sodium coming through the distal tubule, less chloride coming through the distal tubule, that would be picked up by which cell? Which cell of our complex is in the distal tubule? Anyone can remember here? So which cell of our 3 juxtaglomerular complex cells would be detecting this decrease in sodium and decrease in chloride in the distal tubule. The macular denser cells, excellent. So the macular denser cells will detect this and they will help to correct this as well. But this is going to be another stimulus for renin secretion. Okay, so decreased afferent arterial pressure, increased sympathetic activity through the baroreceptor stretch, through decrease in the baroreceptor stretch, really important. So less baroreceptor stretch, more sympathetic activity, and then decrease in sodium and chloride coming through the distal tubule. Okay. And then lastly, how does ANP peptides affect sodium reabsorption? So in short, will it decrease or increase sodium reabsorption? ANP, that is. It will decrease, excellent. So ANP will decrease sodium reabsorption, which is losing more of that sodium in the filtrate in the urine. Okay, so increasing sodium excretion. So be wary of questions that phrase this in different ways. ANP is going to increase sodium excretion, but decrease sodium reabsorption. Same thing with aldosterone. Aldosterone is going to increase sodium reabsorption, but decrease sodium excretion, right? So have a good idea of what's happening as far as if something is being reabsorbed, it's going to be being excreted less, and be wary of questions that may phrase that in different ways. Okay, and then more specifically, the mechanism of ANP there is blocking those apical tubule cells, right? So really understand the mechanism of how these hormones are acting. ANP blocks those apical leak sodium channels, less sodium can get into the cell, and so we can reabsorb less and we excrete more. Okay,